Okay, we're back. And this is starting chapter 4. These are some preliminary chapters, skipping over chapters 2 and 3. Two, um, 2 is over basic chemistry, and you're expected to know that before we come in. And chapter 3, I think, is on cellular respiration and chemical reactions, and you should know that too before you get into A&P. And actually, you should know about cells and cell transport and gene function a little bit also. But I decided to go over the cell chapter. So this chapter 4 is on cells and gene function. So I'm going to do one part on cells, this one right here, and perhaps another video on cells. We'll see how it will take. And then on gene function is going to be another one. So let's start on cells and the cell theory. Now you should write this down because for some reason in the book here on chapter 4, they don't talk about the cell theory. I don't know why, but they should. Cells are a fundamental unit of life. The smallest thing there is that you can say is alive. Like a molecule, like DNA or something, that's not alive. Or the organelles in cells, they're not alive. A cell is alive. So it's a fundamental unit of life, and all living things are composed of cells. Well, there is the next tenet of the cell theory. Fundamental unit of life, all organisms composed of cells. Cells evolved once, about 3.5 to 3.8 BYA billion years ago. How do we know that? Fossils. There's actually microscopic fossils in rocks that have been radioactively dated to 3.5 to 3.8 billion years ago. So this is about a billion years after the planet formed, life started. How did it start? Nobody knows, but it was there. And did it evolve just once? Well, we have one example of cells, and maybe there are other styles of cells that evolved that didn't make it. And this style of cell is the kind that did make it. So that's a speculation also, it only evolved once. But now, all cells come from other cells. You're not going to have any new cells evolving on Earth. And the reason is because we now live in an oxygen environment. It's like 21% oxygen out here. And oxygen is a really dangerous, disruptive molecule that rips molecules apart. It also can destroy cells also. It's very dangerous. We got to breathe this stuff, but it's also a very dangerous molecule. And if the early Earth, like 3.5 billion years ago, there was no oxygen in the atmosphere. And that was real fortunate, because if there had been, I wouldn't be here talking to you. You wouldn't be here, I would be here. No life would have begun on this planet if there was oxygen in the early atmosphere. So since oxygen is here now, and of course it was put on into the atmosphere as the first dangerous toxic gas, probably a couple billion years ago, when photosynthesis was, when photosynthesis evolved, and started putting oxygen in the atmosphere as a waste product of that process. Types of cells. So basically, we got two types of cells on Earth. Prokaryotic cells, which don't have any membrane-bound organelles. They're very tiny, and structurally simpler than the type of cells that make our bodies up. But actually, metabolically, they're much more complex. But anyway, they have no membrane-bound organelles. That is, these structures inside of cells, little factories that do stuff, they're surrounded by membranes, internal membranes. Prokaryotes have no internal membranes, and they're only found in bacteria. That's it. Our type of cell is called eukaryotic cell, and they have membrane-bound organelles such as the nucleus, mitochondria, chloroplast, lysosome. Well, we don't got chloroplast, but plants do. Lysosome, and so on and so forth. That's eukaryotic cells, and that's our type of cell. And all found in all organisms on Earth except bacteria. So there are certain things that are characteristic to all cells, no matter what type they are, what organism they came from. All cells have these things. One of which is a plasma membrane. They're surrounded by a plasma membrane. Right? They have a cytosol. That's what's the internal liquid, intracellular liquid. All cells have that. All cells have at least one molecule, DNA, that is a genetic material for all life on Earth. And all, and there's one organelle that all cells have, called ribosomes. It's not a membrane-bound organelle, so you find in prokaryotes also. And it converts the information that's in on a chromosome into the primary sequence of the protein. So you can think of proteins, they're found in all cells also, they're a requirement. And they're made in ribosomes. So you find ribosomes, it's really the only organelle that you find in every cell on Earth. Okay, the plasma membrane. We'll talk a lot about the plasma membrane because it's really important for delineating the shape and size of the cell and controlling what comes in and what goes out of cells. And that's a big thing. So there's membrane lipids, membrane proteins. Membrane lipids are, for the most part, it's phospholipids. These are little molecules that are shaped like little double lollipops, and they're both polar and non-polar at the same time. And some membranes form what's called a phospholipid bilayer, where the polar part of the phospholipids points to the inside and outside the cells, and the middle part of these what's called non-polar tails, and they help prevent anything from entering and leaving cells, and can separate two aqueous solutions. Just think of it, you can't have, your plasma membrane can't be dissolved by water. Otherwise, the liquid on both sides is an aqueous solution, intracellular fluid, extracellular fluid, both aqueous. Your own membranes will be dissolved. So you have to have a non-polar boundary to delineate the internal aqueous solution from the external aqueous solution and keep them separate because they're very different, not that different in composition, but they're very different in the concentrations of the solute molecules you find on both sides. They're very different between inside and out, and that has to be maintained. Okay, cell size. Okay, if you downloaded the PowerPoint from D2L, this slide isn't on there. I just put it on there. Just realized that it wasn't in there. And it's important. It basically says the same thing as page 106 in the book. Although this is a horizontal scale, that one's a vertical scale, but it's the same thing. It shows you how the sizes of cells. Cells are small. I mean, they're really small. And there's a good reason for that. And it has to do with cell transport. We'll get into that later on, the things that affect the rate of diffusion. Basically, cells are really small to maximize the rate of diffusion. It makes your diffusion, movement of molecules across the membrane is really fast. Okay, so the bottom is starts off with what are called nanometers, which are billions of a meter, then Micrometers is 1,000th of a meter, and then millimeters and so on. So when you get down to prokaryotes, you can see that they're really tiny. Those are the smallest cells there are, the prokaryotic cells. Eukaryotic cells, like red blood cells, well, actually red blood cells really aren't cells. They're called cells, but they have no organelles. So they're not really cells, but okay. Epithelial cells in human egg. Most human cells are too small to see. You can't see, they're too small. And I didn't even realize that cells existed until like the 17th century or something, when the microscope was invented. However, with a human egg cell, if you got really good vision, you can just barely see human egg cell. They don't have plant cells on them, but plant cells are generally a lot larger, but you still can't see them. But what about frog eggs and chicken eggs? Look at this. You can see these. These are really big. That goes against what I just said. Well, it's the frog eggs and chicken eggs, they're not really, the whole thing isn't the egg. Most of a frog egg or a chicken egg is non-cellular material. It's the white of the egg, which is not cellular. It's the yolk of the egg, which is not cellular either. It's a tiny little cell on top of the yolk. It is the actual chicken oocyte, the chicken egg, and it is too small to see. That's true for the unfertilized eggs you buy in the grocery store. Sometimes you can buy fertilized eggs, and you can actually see a tiny little chicken embryo on the surface of the yolk, but then that's lots of cells. That could be like hundreds of cells. Okay, that's true for the frog egg and chicken egg. But then even in our own bodies, you can look at these nerve cells. The cell bodies themselves are microscopic. You can't see a human nerve cell. But there's certain processes of cells that can be variable. Because I'm twiddling my fingers here. How can I do that? Well, there are muscles in your wrist that control that. And there's a nerve, there's two motor neurons, one motor neuron in my brain on this side. It's controlling my right hand. And it's sending information down to your spinal cord. And from the spinal cord, it's a second motor neuron that sends a process way out here and innervates these muscles here. So what is that? That's like a couple feet. That's like 2/3 of a meter or something. So that's macroscopically quite long. And that's an axon. Now, the axon itself is microscopic. You can't see it. But macroscopically, you know it extends out long. So there are some aspects of microscopic cells that are macroscopic. Okay. Then let's get on to the next page, actually. Page 107. Here's a stylized, eukaryotic cell. And what we're going to do is go over these major organelles, these major internal membrane-bounded and sometimes not membrane-bounded organelles. They're, like I said, they're like little factories that do certain things inside the cell. So we're going to look at the nucleus, we're going to look at mitochondria, lysosomes, we're going to look at the rough endoplasmic reticulum, peroxisomes, cilia and flagella, and we're going to look at these, and ribosomes too, and centrosomes and proteasomes. Okay. Plasma membrane. Okay. Oh, I didn't talk about the membrane proteins. I said membrane lipids in one of the previous slides. That is phospholipids make up the major membrane lipid. There's also cholesterol for animal membranes, and we're animals. Cholesterol is an important membrane lipid. Keeps membranes nice and fluid. And then the proteins. There are proteins that are embedded in the membrane, and they do all kinds of important things. when we look at what membrane proteins do, one of the big things they do is cell transport, allow things to enter the cell and leave the cell, and also cell communication. For example, a lot of signaling molecules bind to membrane proteins, and this turns on something inside the cell and tells this receiving cell something to do. Okay. So cell receptors as primary messengers. Cell receptors as receivers of primary message. When we get to the endocrine chapter, we'll learn that water-soluble hormones don't actually enter cells. They bind to some membrane receptors, and they're the primary message. And then they activate internal membranes on the inner side of the membrane that make what's called a second messenger. And this second messenger goes throughout the cell and makes changes. I'm not sure why microvilli and cilia and flagella are there. These are extensions of the cell membrane, cilia and flagella, that allow movement of the cell in one case, and also moving things over the surface of the cell. Cilia, they're like cell hairs. Channel blockers, cystic fibrosis. Particularly when we get into the nervous system, we'll talk a lot about ion channels that allow ions to cross the membrane, because ions are fully charged things and they can't get across a membrane without a membrane protein called an ion channel. And there's and a lot of drugs work by actually blocking certain channels, like calcium channels. Like some people have high blood pressure, have hypertension, take calcium channel blockers, which prevent calcium from entering cells, which actually causes the blood pressure to go down because the heart contracts more weakly. It was less calcium coming into it. Cystic fibrosis it's a chloride channel, a dysfunctional due to a mutation, a chloride channel that doesn't work. It causes this really thick mucus to be produced. So now let's come to the insights. Right. So let's get into membrane transport. Really important topic. All right. Okay, filtration is just pushing something through openings in the cell by pressure being on one side and pressure being less on the other side. So it's like a sieve. You're pushing things through holes. And mostly we'll see this in the next semester. We get into the urinary system. We'll see there's a filtration process going on in nephrons. Diffusion. Diffusion is a kind of what's called passive transport. It's the movement of molecules from an area of high concentration to an area of lower concentration. Right? This is called net diffusion. And you need two things for net diffusion. One thing, you need a permeable membrane. The membrane has to be permeable to this substance, or you're not going to have diffusion. Then you have to have a concentration gradient. Means the concentration on one side has to be larger than on the other side. And it'll go from where it's highly concentrated to where it's more lowly concentrated to try to reach what's called equilibrium, which is the same concentration of this substance on both sides of the membrane. Okay, oftentimes when we talk about diffusion, we talk about diffusion of solutes, things that are dissolved in water. When water is the biological solvent, and body fluids are complex solutions. where water is the solvent, and there are things dissolved. And these things might or might not cross the membrane. But water is also a molecule. And can it cross the membrane? Well, for almost all the cells in your body, this is the case. Water crosses the membrane. Just follows its concentration gradient. And this is called osmosis. So it's simple diffusion, but it's the diffusion of the solvent, not the solute. So it gets a special name, osmosis. And it's very important to maintain what's called the osmolarity of your body fluids. Because all of your cells, except for some specialized ones in your kidney, are permeable to water. So it can be dangerous if this is part of the water balance of your body, to make sure you don't have too much water or too little water in your body. This is called tonicity. It's the measure about how much things are dissolved in your body fluid and how much water is there. There's a ratio there. It's called osmolarity, and it has to always remain the same. It's a homeostatic variable, the tonicity of your body fluids. And it basically comes down to the water concentration. The more things that are dissolved in an aqueous solution, the lower the water concentration. The fewer things that are dissolved in water, the higher the water concentration. Carrier-mediated transport mechanisms. Now we get into not just molecules just simply diffusing across membranes, but being assisted across. And sometimes without the need of cellular energy, sometimes with the use of cellular energy. And its specificity. You know, it's true even for simple diffusion with ion channels. You know, there are sodium ion channels, potassium ion channels, calcium ion channels, and they're specific. And particularly carrier-mediated, they're very specific. There's glucose transport proteins that only glucose binds to. Right? And they can be saturated. That is, if there's really high concentration of this stuff, you can overwhelm the ability of these carrier proteins to bring stuff in or push stuff out. Uniport, symport, antiport. This is what's called secondary active transport that uses the sodium concentration gradient. Sodium is always more concentrated outside cells than inside. And you can use that to bring things into cells, what's called symport, or push things out of cells called antiport. Don't worry about the uniport. To symport and antiport. Facilitate diffusion. This is just helping a molecule cross the membrane. Glucose has to be helped. It has to bind to a glucose transport protein and help across the membrane. You can't just diffuse. And that's called facilitated diffusion. But it follows a concentration gradient. Glucose has to be more concentrated outside of cells than inside in order for it to come in. And that's always the case, as a matter of fact. Active transport. You're using cellular energy, ATP, to bring something into a cell or push something out of a cell against the concentration gradient. That is from an area of low concentration to an area of high concentration. If you want to do that, you got to use ATP, cellular energy, to do that. That's called active transport. Bulk or vesicular transport. You can think that this is a different kind of active transport, where you bring in or push out really big things, mostly like proteins or protein complexes. You take them in, you push them out. It has to do with, it's called vesicular transport because you have to form these vesicles, these membrane balloons that either can bring stuff in a process called endocytosis, or these membrane bubbles on the inside cell merging with the cell membrane to push things out, and that's called exocytosis. Inside familiar hypercholesterolemia, that is LDLs, the bad form of cholesterol. This is brought into cells by vesicular transport. And these LDLs bind to LDL receptors on the cell. And this causes the membrane to invaginate and bring these LDLs inside the cells. That's good because it gets it out of the blood. The LDLs are a direct risk factor for heart disease, and it's good to bring them into cells. Familiar hypercholesterolemia is that you have one of the genes that codes for the LDL receptor is defective. So half of your LDL receptors don't work, and you can't nearly bring in as much LDLs as you should be able to. And you're going to be in really high risk for heart attack in your 40s, maybe early 50s. That is if you have one defective allele for the LDL receptor. You have two defective alleles, you're going to have a heart attack in your teen years. So this is what the statin drugs are for. Even though I take a statin drug, they're used to lower cholesterol, but mostly they're most important for people who have this genetic disease of having defective LDL receptors. Okay, here's why tonicity, or the osmolarity of your cells, is important to maintain cell size. What we want to do is make sure that your body fluids are isotonic to the intracellular fluid in your cells. What that means is the water concentration is the same on both sides. And we'll learn that for most things, the concentration of stuff outside cells is very different than the concentration of stuff inside cells. It's really different, has to remain that way. A couple things are not different. That is water concentration. That has to be the same on both sides. And concentration of hydrogen ions and hydroxide ions. That has to be the same on both sides. Everything else is actually different. So if the concentration of water on both sides of the membrane is the same, then water crosses the membrane, but every time a water molecule enters the cell, one will leave, and the water concentration won't change. There's two things that could screw that up, and that is too much water on the outside of your cells. This would be a hypotonic solution. If your blood or interstitial fluid becomes hypotonic to your cells, that means as water concentration is higher on the outside of cells than the inside, water will rush into cells and swell cells up. And if it's hypotonic enough, it will burst cells. And this is a condition called hypernatremia, hyponatremia. It actually refers to too little sodium inside, but it's better to look at it from the water aspect. So actually, I often say, can you kill yourself by drinking too much water? And I don't mean drowning, I'm just drinking and stuff. And the answer is yes, you can. And it happens sometimes because people drink so much water, their brain cells swell up. Your kidneys are the primary responsibility of maintaining this osmolarity, but they can only work so fast. So if you drink faster, then your kidneys can get rid of excess water. Your cells will swell up and burst, and you'll be dead. So water, just like anything else, you take enough of a substance, it'll kill you. It's really hard to do because, if you have way too much water, your body starts to tell you that because like nauseating to think of even drinking any more water. And that's a defense mechanism to tell you stop drinking water. The opposite is dehydration. Most people think, that that's the more common thing. It probably is the more common thing. You don't drink enough water. the water concentration drops outside of cells. This will suck water out of cells and cause them to shrink. And again, in severe dehydration, it can shrink so much that they'll be killed. So that can be lethal situation also. The strange thing is that if you're overhydrated or underhydrated, the symptoms are exactly the same. Like nausea, tiredness, confusion, same thing. So you can't tell if you get those symptoms, which state you're in. So you listen to your thirst center in your brain. They'll tell you, if you're dehydrated, you're gonna be really thirsty. Drink something. If you're overhydrated, the thought of water is going to be like nauseating to you, and that tells you you're overhydrated. Okay. This is new also. I just put this one on up. This is all these, and it looks pretty complicated. Well, it isn't. This is on page 110 about all the different types of membrane transport. Passive processes that don't require cellular energy, and active processes that require the expenditure of cellular energy. And of course, that cellular energy is ATP. So diffusion osmosis, passive, completely passive. And diffusion becomes simple diffusion and facilitated diffusion. Now, this is kind of misleading. It says simple diffusion, no transport protein required. Not necessarily. You don't necessarily have a transport protein, particularly if you have a small non-polar molecule. It comes across without anything. It goes right through the phospholipid bilayer. But you got to bring ions in and get rid of ions. And they don't cross the membrane without a protein. Now, without an ion channel. So simple diffusion may, of certain things that are charged molecules, that requires a membrane protein, right? Even though it's simple diffusion. So don't get the wrong impression that all simple diffusion doesn't require membrane protein. Only simple diffusion of non-polar molecules doesn't require. If you get a polar molecule, even water requires these proteins called aquaporins to be in a membrane. So it's kind of misleading there. All right. Then facilitated diffusion is channel mediated. Ions move through channels and carrier mediated. So what they're calling the movement of ions here, channel mediated. So it's simple diffusion, but it's channel mediated. So I should realize this. They're going to call it, so when they have simple diffusion on this chart, they're just referring to nonpolar. If it's they're calling the movement of ions through ion channels facilitate diffusion. Most books don't do this. Most biology books don't do this. They just call it simple diffusion, but with a protein needed. So this book is a little odd in that way. Carrier mediated, small polar molecules moved by carrier protein. That's like glucose and amino acids. They require carrier protein. Okay. Active process require expenditure of cellular energy. Active transport, move the ions or small molecules moved against the concentration gradient. Primary active transport energy source directly from ATP. Secondary active transport from movement energy source from movement of another substance, and that is the sodium concentration gradient that can be tapped to bring things into cells by symport, push things push things out of cells by antiport. Then in vesicular transport, exocytosis, pushing things out of cells with vesicles merging with the cell membrane. Endocytosis, vesicles form as materials are brought into cells. And this is phagocytosis, cell eating. Right? And pilocytosis, cellular drinking, and receptor-mediated endocytosis, where a receptor is required in order to form this vesicle to bring this thing in. And an example of that, we'll remember, were the LDL receptors. Okay, on this part, I'm going to call it. It's been about a half an hour, and this will be 203 cells 1. Okay, so I'm going to stop recording now, and I'll pick you up with the next video. Where is it?